Hi everyone. So the purpose of this video is to just re-explain the content in the gene expression PowerPoint so that if you're still unsure about any of the aspects of this part of the DNA proteins topic and want to listen to it again, you've got this recording. So you can listen to it as many times as you like. So before we get into the details, one thing that I'll just clarify from the science understanding is that the arrows are basically the sort of main ideas that you have to understand, and the dot points are sort of skills and things that you actually have to be able to do. So if we look at that in the context of gene expression, you can see that ideas like the fact that the phenotypic expression of genes depends on factors controlling transcription and translation is just a concept that you have to understand. So that means that you're unlikely to be asked to specifically describe, how transcription and translation are regulated. Whereas if we look at the DNA methylation, you can see that it says here in this dot point, recognize that cytosine nucleotides in DNA can be methylated and this alters gene expression. And you have to explain how epigenetic modifications in genes that control cell division, such as changes in DNA methylation, can lead to cancer. So ultimately, when you're revising this bit of the topic for the test and for the exam, they're the bits that you really have to be able to explain and describe really well. So other aspects such as the concept of, well, there are these factors that control transcription and translation is just an idea that you have to have a broad understanding of. Okay, so if you're still a bit unsure about the difference between those two parts of the subject outline, let me know and we can go through it more in class. So to start off with, what is gene expression? So we've talked about, what genes are and the fact that genes are read and transcribed into a condensed copy, which is the mRNA, and that is what is read to produce the protein in translation. So ultimately, what determines when that happens is a really complex process. And so when we look at it, we're generalizing and we're really simplifying it. But when we use the word gene expression, what we mean is the protein product that gene encodes is being produced. So gene expression means that transcription is occurring and translation is occurring. So we're getting that specific protein product. So the question then is, well, like, how do cells know? So that's where we have regulators that decide when the gene is switched on, when the gene is switched off, and how much of that protein is actually being made. So before we get into some of the environmental factors that can influence that, we just have to sort of go over a bit of revision about chromosomes. So remember that all cells have 46 chromosomes. They have 23 pairs. One of those pairs are called the sex chromosomes. So that's the XX in a female, XY in a male. Any chromosome that is not a sex chromosome is called an autosome. So in body cells, we have 22 pairs of autosomes and then 1 pair of sex chromosomes. So chromosomes have specific genes on them at specific locations. Each chromosome has 1 copy. So that means that if you have 2 copies of the chromosome in a cell, you have 2 copies of each of those genes. Different variants of those genes are called alleles. So for example, we have the gene for eye color, but you could have the blue eye color version, you could have the brown eye color version, you could have the green, and so they are the alleles. So what genotype means is genotype is the genetic makeup. So in other words, what alleles, what variants of that gene does the individual have? What 2 copies of the gene do they have? That's what the genotype is. The phenotype is the outward appearance. So it's how the organism looks, it's how the organism acts and how it feels. So the phenotype is determined by the genotype. So if we have a look at the diagram on this slide, we've got a gene for a tall plant and an allele, for the dwarf plant. So which alleles the offspring inherits determines its phenotype, its characteristic. If it inherits the alleles for tall, it'll be a tall plant. If it inherits the characteristics, the allele for the dwarf, it'll be a short plant. So the genotype determines the phenotype. So when we talk about the phenotypic expression of a gene, what we mean is, well, what physical, biochemical, or physiological characteristics does that gene produce? So for example, if you look at the two alleles for eye color that someone has, what physical characteristic does that produce? What color are their eyes? Are they brown-eyed? Are they blue-eyed? It could be biochemical, so it could be, the alleles, the genotype that determines what blood group somebody is, or their ability to produce insulin, for example. And so there are environmental factors that can influence the genotype, which therefore influences the phenotype. So let's look at a few examples of that. So here are some examples of gene products that influence phenotypic expression. So for example, if someone has a gene product that leads to a lack of insulin, the phenotypic expression of that is that they're a diabetic. Or, if someone has a lot of estrogen being expressed and producing estrogen, that means they're going to develop female secondary sex characteristics. So again, those characteristics are the phenotype. The gene product is the estrogen gene. So some of the environmental factors that influence gene expression include gender, chemicals, temperature, and light. So I'm going to go through this bit pretty quickly. If you have a look in your note set, you have quite a lot of detail about some of these examples of how environmental factors influence gene expression. And it starts on page 31 of your notes and continues until page 33. Okay, so if we start with gender, there are some genes that are expressed differently in males and females, so they're sex influenced. So what that means is that both males and females have the genes. So for example, the genes for lactation, which is the production of milk. However, those genes are only expressed in females. So again, expressed means only females will transcribe and translate that gene to produce the proteins that lead to lactation. Males will have those genes, but they will not be expressed. Another example is baldness. So the hormones testosterone and dihydrotestosterone both influence baldness. So because those two hormone levels are higher in males, they're more likely to go bald. Chemicals can also have an influence. So we've looked in class in a lot of depth at thalidomide. So thalidomide was a drug that was prescribed for insomnia. It was used for nausea. It was used as an anti-anxiety medication, and what they didn't realize early on in its use, that if it was taken at a particular stage of pregnancy, it impacted the expression of genes involved in limb formation. So babies were born with severe physical disabilities because they weren't able to form normal limbs. So the presence of chemicals and drugs in the organism's environment can have a big impact on gene expression as well. Lastly are temperature and light. So these are just two examples of animals where temperature influences gene expression. So for example, in the development of a species of turtle, they're very sensitive to temperature at a particular period of development, which basically, depending on what temperature they are exposed to, influences the gender of the offspring. Similarly, with this species of Himalayan rabbit, if they are reared at 20 degrees or less, they develop the darker pigmentation in the ears and the nose and the feet and the tails, whereas if they're reared above 30 degrees, they're not. So temperature influences whether certain genes are expressed or not, which changes their phenotype. They look different. Okay, so we're just going to link this whole concept of, genes being expressed in some cells, or, one gender and not the other, in the context of cell differentiation and cell stem cells. So if we just revise a bit from year 11, hopefully you'll remember, well, the cell is the basic unit of life. If we have a group of cells working together, we call them a tissue. And a group of tissues working together is an organ. And then when you have groups of organs working together, we call that an organ system. And then an organism is any living thing made of one or more cells. So if it's made of multiple cells, if it's a multicellular organism, we have these tissues, organs, and organ systems all working together. So how do we start from an identical cell and end up with a wide variety of different cell types. And so that is due to, again, gene expression. So in sexual reproduction, we call the sex cells, so sperm and egg, gametes. Okay, so gametes contain half the normal number of chromosomes, and that's so that when you fertilize the egg, so when you get the fusion of the genetic material from the sperm and the egg, you restore the normal number of chromosomes. So the egg has a single copy of each, has 23. The sperm has a single copy of each, it has 23. And so then when they fuse together and you have a fertilized egg, it has the correct number. It has a total of 46 and it has 2 copies of each chromosome. So that initial fertilized egg is called a zygote, okay? And so what happens is once you have that zygote, it undergoes a rapid series of divisions. And so when we use the term an embryo, all an embryo is a cluster of genetically identical cells that derive from that original fertilized egg. And so the cells in that embryo aren't specialized. So they have all of the genetic information. They have the potential to become any cell type. So we call them stem cells. So what tells them to become a skin cell or a neuron or a blood cell is the genes that are expressed and the genes that are silenced. Okay, so if we have a look at this diagram at the top, we have these 2 identical daughter cells. In one, neuron-specific genes are expressed. So again, expressed means those genes are the ones that are transcribed. They're the genes that are translated, giving rise to a neuron. Whereas in this other cell, which is identical genetically, we're expressing different genes. So we're transcribing and translating different genes, and therefore this daughter cell is becoming an epithelial cell. So again, just to reiterate, all of those cells have a copy of every single gene. So genetically, they are identical. What makes them different is the genes that are being expressed and the genes that aren't being expressed. And so we call that process of, genes selectively being expressed or not as cell differentiation. So it really leads to cells becoming specialists at making certain proteins. And so the proteins that they make determines the structure and function of the cell. So if we have a look at this diagram here, it just illustrates the fact that different cells have shapes and structures that help them carry out their function, and there's a really wide variety of structures and functions of cells. So for example, a nerve cell is a neuron is transmitting information, from the central nervous system to the rest of the body or back. And so these cells can be really long. They can be up to 2 meters long, and they have this long structure called the axon, which allows them to transmit that information. If we have a look at the sperm cell, they have to be able to move quite a long distance, and because they're competing with each other, they have to move quite quickly. So they have a long flagella that enables them to do that, to help them to swim towards the egg cell. So all cells are highly specialized, and the reason why they're highly specialized is because of the genes that they express. Okay, so if gene expression is referring to that process of, when the protein is made and how much of the protein is made via transcription and translation, gene regulation is the processes that control that. Okay, so it's just a general label. So because protein synthesis, gene expression occurs at various stages, we could control how much protein is produced by influencing transcription. We can influence it by regulating translation. It could be how the introns are spliced out after transcription. It could be modifications to the DNA. So again, we'll start by going through 1 and 2 here, but they are like general ideas that you have to understand and, be able to explain. The one where you really have to be able to justify it and go into more detail is DNA methylation. So of this section, the most important part to understand well and to be able to answer questions about is number 3, which is DNA methylation. Okay, so again, like I said at the start, this is a very complex process. So we're oversimplifying it just to help you get the general idea. So because you've got a copy of the PowerPoint, I'm going to do much of my explaining with the diagrams. So you can be looking at the picture while I explain it, and then you can open the PowerPoint if you want to look back at the slides that have the actual writing on them. So if we skip ahead to this one, you'll see here that we've got a dark blue section of the DNA, and that is the gene. So that's the bit of the DNA that codes for the mRNA that codes for the protein. And so right before the transcription start point is a specific sequence in the DNA called a promoter. Okay, so the promoter is part of the DNA. It's right before the gene where transcription starts. So for transcription to start, RNA polymerase has to be able to access the promoter. So RNA polymerase can't read the DNA template strand. It can't add the RNA nucleotides to synthesize mRNA if it can't access the promoter. So how does it access the promoter? What starts that process of transcription? So there are proteins called activators. And so basically, and if we're going to be more specific, this diagram's being a bit more specific. It's talking about transcription factors, but basically activators bind to the promoter. And what that does is it allows the DNA to uncoil further so that RNA polymerase can get in and start transcription. So activators are what initiate that process of transcription. They bind to the promoter, they expose the DNA so that RNA polymerase can get in there, bind to the promoter and start transcription. We also have gene products called repressors that do the opposite. So if we have a look at this diagram, repressors bind to the promoter as well, but what they do is they prevent RNA polymerase from attaching. So they basically get in the way. So if the repressor's on the promoter, the DNA is more tightly coiled, RNA polymerase can't access the promoter, and so therefore transcription doesn't occur. Okay, so takeaway message from this is we have gene products, we have proteins called activators that bind to the promoter, enable RNA polymerase to bind, which starts transcription, and we have other gene products that are proteins called repressors that bind to the promoter that prevent RNA polymerase from binding, which therefore block transcription. That's all you need to know. If you understand that main idea, fantastic. So another way we can regulate gene expression and the production of the protein is through alternative splicing. So we talked in transcription about the fact that initially the whole gene is transcribed, which includes these non-coding regions called introns. The parts of the gene that actually code for the protein are called the exons. So after transcription, those non-coding introns are spliced out, and it's that condensed mature mRNA that moves out of the nucleus. So the way in which the introns are cut out and how the exons are combined influences ultimately the protein sequence. So all alternative splicing means is that the way in which the exons are combined can give rise to different variants of the protein. And those variants are called isoforms. And again, it's much more complex than that, but if you understand the key points that are on this slide, that's all you need to know about alternative splicing. Wouldn't worry about anything else, just know, okay, you can combine the different exons, gives rise to different protein variants. We call those isoforms. The last way we can regulate expression of a gene is by influencing translation. So translation cannot occur without the ribosome. It is the ribosome that moves along the mRNA and is the platform for the mRNA and the tRNA binding together. Now the ribosome is actually made up of two subunits, which a subunit is just like two parts, two pieces of a puzzle. So those two subunits have to come together to form the ribosome for translation. Now one of the ribosome subunits, which is called 40S, you don't need to know that, but it has this protein on it called initiation factor EIF2. And pretty much if that initiation factor is phosphorylated, which means you just add a phosphate group, it changes the shape of that ribosome subunit. So if you change the shape of one of the two pieces of the ribosome, they don't fit together. They don't fit together, they can't assemble on the mRNA, there's no translation. Okay, so again, you just have to understand the main idea here, which is if you block the assembly of the ribosome, you prevent translation from occurring, which means that that gene is not getting expressed. You're transcribing it, you're getting mRNA, but the mRNA is not getting read to produce a protein. So just to summarize some of those different points in the process at which we can regulate gene expression, it can be during transcription, it can be after transcription, which involves alternative splicing, it can be during translation, it can be after translation. Okay, so there's multiple points at which we can influence whether the protein is produced at all and how much of the protein is actually produced. So this last bit, talking about epigenetics and DNA methylation, is the most important bit. This is the bit that you tend to get examined on. This is the bit that you have to be comfortable explaining for 4 marks. Okay, so basically we've talked a lot about genes and the influence of the gene sequence, but the DNA and the histones, so the proteins around which it is coiled, can also influence gene expression. So chemical modifications that are on the DNA and are on the histones are called the epigenome. Okay, so it's how the DNA is being packaged around the histones. Now the epigenome, whether the DNA and histones have those chemical modifications and where they are, is influenced by the environment. Okay, and so it's this concept which explains the fact that you can have identical twins who have some differences in their characteristics and their phenotype, or one develops cancer and one doesn't. Okay, and as we understand more about the epigenome, it's explaining more about why you can have two individuals who are genetically identical, but you can get differences in gene expression, and so it's to do with these chemical modifications of the DNA and the histones called the epigenome. So basically these chemical tags, this epigenome, can do one of two things. It can cause the DNA to coil more tightly around the histones, which essentially switches them off or silences them, because if they're tightly coiled, they can't act as a template. RNA polymerase can't get in there to start transcription. Or maybe it uncoils sets of genes, so it activates them, it exposes them, RNA polymerase can get into that promoter. So some of the signals that influence these epigenetic chemicals are things like our diet, stress, and physical activity. So for example, there's been studies showing, people who regularly eat foods like broccoli and garlic activate genes that are protective against cancer. So here are some other examples of environmental factors that have been shown to influence the epigenome and therefore transcription and translation. So a lack of oxygen because of high altitude, so being up, can increase red blood cell production. Increased UV exposure can lead to a change in skin color. Increased light intensity can lead to an increase in plant growth. So these environmental factors influencing phenotype is as a result of their influence on transcription and translation. So this is just showing you the DNA being coiled around the histones, and so if you have a look at the box with the histone tails, so sometimes like acetyl groups can be added to the histone. Okay, so you can see that having that chemical tag there means that it can't coil as tightly, which makes it more accessible, so that those genes are going to be more likely to be activated. If you add a methyl group to the DNA, that causes the DNA to be more tightly wound around the histone, which makes that DNA inaccessible to RNA polymerase. So therefore, the gene is inactive. Okay, so this is a more sort of zoomed in specific. So DNA methylation, what that means is you have a methyl group that's added to the DNA strand. It's usually added to the cytosine base. So think you've got your DNA strand, you've got your nucleotides, and some of those cytosine bases have a methyl group added to them. Okay, generally is to the DNA to the cytosine base, but it can also be to the histone proteins. So what happens is the DNA is more tightly coiled, and so it blocks the action of RNA polymerase. So if the DNA is methylated, it has these methyl groups on cytosine bases, it essentially can lock genes off. Okay, so more tightly coiled, inaccessible, so the gene is inactive because RNA polymerase can't bind to the promoter, can't initiate transcription. So this is just showing you again. So the line with the arrow, that's basically our DNA strand. So we've got no methylation, transcription factors combine, you activate the gene, you get gene expression. The methylated DNA on the cytosine reduces binding of the transcription factors, which means the gene is inactive, and so you don't get gene expression. So I think this is another just really nice diagram explaining what happens. So DNA methylation effectively silences the gene, and it just shows you there how it results in this tight coiling around the histones, which is why that happens. Whereas an acetyl group tends to cause it to be more loosely coiled, so the gene is on. Now, even though the epigenome involves the addition of acetyl groups and methyl groups, from the subject outline, the only one you have to be able to explain is the methyl groups. Okay, so while these diagrams have the acetyl groups as well, that bit's not examinable. So you just have to know number one in this diagram, which is the DNA methylation. So there is increasing amounts of evidence to suggest that there are many diseases that are related to these epigenetic changes. So in other words, if the DNA is over-methylated, it prevents like healthy regulation of genes. So things like diet, stress, exposure to cigarette smoke, heavy metals and other chemicals can change these patterns of DNA methylation. And some of the diseases that have been linked to these epigenetic changes include some autoimmune diseases, cardiovascular disease, and cancer. So cancer is uncontrolled cell division. So normally you have these genes that produce proteins that regulate when the cell divides, that regulate the repair of DNA. And so if you change the sequence of any of those genes, which we call mutation, you get abnormal proteins, which means that cell division happens in an unregulated manner. This is something that we'll come back to in the cells topic, at which time we'll look at it in a lot more detail. So for now, we're just touching on it from the perspective of epigenetics. The uncontrolled cell division in the cell cycle bit is something that we will come back to. So don't worry too much about the intricacies of that just yet. But basically, there are certain genes that, basically prevent the cell from dividing uncontrollably. And so if you methylate that gene, it's switched off, which means that the cell can divide uncontrollably and can produce excessive numbers of new cells. Similarly, if you have a gene that normally repairs DNA, If that's silenced, you're going to get less repair of DNA, which means you accumulate more mutations, which means that the cell is more and more likely to become abnormal and to become a cancer cell and to lead to disease. So these are just a couple of examples. These diagrams are more specific than you have to know, but just show you how various genes that regulate a cell's behavior in terms of it being able to repair its DNA, in terms of it being its division being controlled. If those genes are methylated or not, it can therefore impact on the behavior of the cell and how likely it is to become a cancer cell. Because if the genes being silenced are ones that regulate DNA repair, that regulate cell division, well, then it can lead to cancer. So in summary, cells are specialized in structure to perform a specific function. They're all genetically identical. What leads to that specialization is the genes that are expressed. So in other words, which genes are activated and transcribed and translated and which ones are not. Okay. There are a variety of mechanisms for switching genes on and off. It can be mechanisms in transcription. It can be in alternative splicing. It can be during translation, it can be after translation, and there are also environmental factors that can alter gene expression, whether it's chemicals, whether it's temperature, whether it's gender, whether it's chemical modifications to the epigenome as a result of diet or exercise or stress. All of these things influence gene expression, which impacts the function of the cell and therefore its behavior.